What's up, my deformed dendrites and aberrant axons? Welcome back to the Nerve Damage Podcast. On this episode of Neuro News, our special guests are here to discuss microcephaly and investigate the possible role that radioglia have in this disease. Today, we have Anna, Nick, Ewan, Bowen in the studio today. Everyone give a round of applause and introduce yourself.
Hi.
Hello there.
What's up?
Howdy.
All right, everyone. So let's just jump right into it. In my last podcast episode, we spoke about the Zika virus and how one effect it can lead to is a disease that makes babies' heads smaller at birth. That's called microcephaly. Do you think you all could give us some information about this disease as well as who it affects and whether or not we should be worried about it?
Yeah, I thought it might be helpful to begin with a case about microcephaly. Michael is a 10-year-old boy from Ohio. During his pregnancy, Michael's mother, Charlotte, felt fine. There were some early signs during prenatal ultrasound appointments that something might be a little abnormal, but it was difficult to diagnose. Michael's parents had struggled to get pregnant and decided to keep the pregnancy. The delivery was relatively standard with no significant complications. After birth, however, Michael's doctors and nurses noticed that his head seemed a little smaller than was to be expected. They measured his head circumference and found that he was in the second percentile. This was enough to diagnose him with microcephaly.
It sounds like a difficult situation. How old is Michael?
Well, now Michael is 10 years old. His parents have dedicated a lot of energy and resources to caring for him. Michael attends a school specializing in providing education for students with special needs. He has special aid at school and someone who takes care of him at home after school and on the weekends. Outside of school, Michael enjoys playing soccer and baseball in the afternoons.
It seems that Michael's getting the support he needs. Does Michael have any other impairments?
Michael sometimes has trouble controlling his emotions and can physically lash out. He has other cognitive delays as well. Both of his parents emphasize the amount of attention, care, and resources that Michael needs.
The thought of not having someone who can take over when I need a break or you need a break, I can't speak to that enough, the importance of that.
Before becoming pregnant with Michael's sister, Kate, they discovered they were carriers for a specific gene known to cause microcephaly. Michael would have received the affected genes from both his parents, resulting in his condition. I want to emphasize that Michael is a happy and healthy kid with microcephaly. It does take a lot of resources and extra care, and his family is fortunate enough to be able to provide them for him.
Anna, thank you so much for sharing this information with the audience today. Most of us can say that this is our first time hearing about this disease. Something I want to add, Bowen, do you think you could tell our audience more about microcephaly?
Sure. Microcephaly is a clinical diagnosis made at or after birth that describes a small head and brain. Individuals born with this disease still have all regions of the brain, but the regions are made up of less cells. One should know that even though some children with microcephaly are healthy, children with microcephaly like Michael suffer from serious cognitive and behavioral issues. Furthermore, even though roughly 1 in 1,000 children is born with microcephaly in the US, its broad implications relating to the Zika virus are worth noting. One of the most medically challenging aspects of microcephaly is its diagnosis. A fetus can exhibit abnormalities related to microcephaly late in the second or third term of pregnancy. Because of signs of the diseases present late in pregnancy, parents are faced with particularly challenging decisions regarding their family's future. In Michael's case, he could participate in special needs school and sports. It is essential to raise broad awareness of microcephaly so that more children like Michael can be helped. Nick, can you talk more about what causes microcephaly exactly? What happened in the cellular levels that can so greatly affect the behaviors?
Yeah, of course, Bowen. We wanted to discuss a specific type of cell, which is called radioglia. Radioglia are stem cells, and stem cells are a kind of cell that can divide to form many different kinds of cells. Radioglia were initially considered to be only structural support cells in the developing brain. However, research from the Department of Neurobiology at Columbia and at UCF suggests that radioglia may play more of a role than just structural support. These cells have a second critical function. They can act as neuroprogenitors, the cells that create neurons. So, radioglia do act as structural support for the brain, but they also, excitingly, help the brain develop neurons. This new finding is relevant to research on microcephaly because neuroprogenitors are reduced in the brains of affected patients.
Oh, okay. Yeah, no, I got it now. Makes sense. One question, Nick. How did the researchers determine this new function?
They injected a retrovirus which is a virus that targets the cell's specific DNA to label those cells.
Wait. Hold on. Nick, you're confusing me and the audience. You're speaking another language here. I don't know what you're talking about. What is the retrovirus and what does it have to do with DNA? I'm sorry they have to ask me these questions, but I have a smooth brain. There's not that many faults in my brain.
Fair questions. DNA is the information that makes up our genes. A good analogy is that DNA is our body's recipe to function correctly. The retrovirus inserts its information into the cell's DNA, so the cell can now follow the recipe of the retrovirus, the DNA, and produce what that recipe calls for. In this case, the retrovirus's recipe tells the cell to produce a fluorescent molecule that makes it visible to researchers. Researchers can then track the location of specific cells with the retrovirus DNA by following the fluorescent molecule. These are the radioglia in this study. So they follow the location and determine the identity of the fluorescent cells and their progeny. Progeny is the cells that are produced from the original cells. So think of it as the offspring of the cells. They noticed that some fluorescent cells were neurons, proving they came from fluorescent radioglia.
Okay, I think I get you, but I'm still confused. So how exactly are these cells relevant to diseases like microcephaly?
Well, when everything works correctly, radioglia function to develop the layers of our brains by helping neurons migrate to newly developing regions and producing neurons themselves. Research shows that maintaining regular neural progenitor number is necessary to develop a normal size and normal functioning brain. As Nick discussed, microcephaly causes small head development and is generally associated with undeveloped brains. This means that microcephaly may result from insufficient neural progenitors or improper migration and proliferation of neurons.
Okay, I'm still confused on how radioglia help neurons migrate to other parts of the brain. Could you explain that?
Some promising research from the Walsh Lab at Harvard Medical School ties together one of the common genetic causes of microcephaly with brain development. One of the most common causes of microcephaly is having two non-functional copies of the ASPM gene. The Walsh Lab works in a mouse model to find the relationships between neuroprogenitor activity and brain size. The ASPM gene facilitates the production of neuroprogenitors like radioglia, which in turn lead to healthy brain development. ASPM increases the production of basal radioglia, a particular kind of neuroprogenitor associated with increased neuron production relative to other kinds of radioglia.
So kids like Michael, they inherit two faulty copies of the ASPM gene from their parents. With no active ASPM gene, there is no production of that helpful radioglia and therefore a smaller brain size. Because the ASPM gene is explicitly involved in growing the cortex, this explains why the architecture of the brain, the placement of things, the general structure, is the same for people with microcephaly, but the outer areas, the cortex, are smaller.
So, to address your question about treating microcephaly, if we can figure out how to promote an increase in radioglia levels in a fetus with microcephaly, maybe we can encourage normal development of the brain.
Okay, that's interesting. One thing I want to add, from the looks of it, I can tell that like my point of view, as well as the audience's point of view, has definitely broadened during this talk from all the information that you all brought to the table. You spoke about microcephaly's impact on the parents and children's livelihood during this talk, and you've also discussed radioglia and how they relate to microcephaly. One question I do want to ask is, how can this information be used for other areas of research?
Well, one of the fascinating takeaways I had from the research we discussed was there's potential for us to facilitate the repair of damaged brain tissue and to treat brain injuries and disorders. Understanding the role of radioglia in forming new neurons and how these neurons then migrate to the correct location of the brain has important implications for how we will address medical and scientific questions going forward.
Yeah, the finding that radioglia can form new neurons and the recent finding that radioglia activity impacts brain size suggests a therapeutic potential. Whether this is possible as a new neonatal treatment for fetus with microcephaly or treatment for someone later in life with neurodegenerative diseases is yet to be determined. There's so much to learn, and we hope to continue to draw connections between essential neuroscience components and real-world consequences like microcephaly.
Wow, that was well spoken from all of you. Unfortunately, time is coming short on this podcast, but thank you all for coming in and talking to me on this episode of the podcast. Coming up next week for our audience, we're going to be talking about sleep, What is it? Why is it important? And what happens when we don't get enough of it? I hope you all learned something from these folks today. And something I want to add is that don't forget to like, comment, and subscribe to the Nerve Damage Podcast. And don't forget to support us on Patreon because we do not get enough ad revenue on YouTube. With that being said, have a wonderful rest of your day.
Bye.
Goodbye.
Bye.